No, it's really a pleasure, Rector Miller. Thank you for inviting me, and Chris, thank you as well. It's an honor to be here and to speak about what I think I learned over my career, which has been interesting because I was in academia. I was mostly from the physical sciences part of the world. You know, I trained as a physicist, mathematics, bioengineer, and then doctor, and MD. Medical doctor, and I try to combine the two, and I think that was the key really to understand what the future was for me, quantitation. And I think, as we will discuss it, that has helped me academically. And then, obviously, doing that, I was able to, take the position of NIH director for 7 years. And then after that, I was in industry. And so today, what I would like to do is really share with you ideas and concepts that have come to me from this unusual pathway and career. So what I'd like to do is really talk to you about what I learned about the missions and visions of academia, government, and industry. And I will tell you things you already know. But I think it's a good idea to place that in the background of the pillars, if you will, that drive every one of these entities, because they interact with each other and they form an ecosystem. They have different missions, but they're complementary to each other. I will also talk to you about what I think I know best, and that is a vision of global science and technology, worldwide. Because it's really hard to know where you are unless you understand the total picture of science and technology. And I'll try to focus a little bit on Austria as I did my research for this talk. And then I will talk about, obviously, within this large context, where is biomedicine? Where is biomedical research and development? Where are the emerging trends and what I consider the most important trends that I think every one of us here should really pay attention to? And obviously, being from the imaging sciences, I'll give you some examples of what I see in imaging that may have a role in this new vision of P4 medicine as he described it. So, key missions and key drivers. What is academia? What do we do in academia? So when you look at academia, first and foremost, in my view, is that academia for any country is the guardian of the intellectual heritage of the society. No one else is. And the second goal is that you transmit that knowledge. And it is a strong evolutionary force to be able to transmit from generation to generation knowledge and new knowledge at the same time. It forms human capital. Of all the strengths of any society, the human strength, the human capital is the key. And so the universities and the academic system is responsible for creating that capital. Obviously, universities have been a social escalator. They are a factor of social change and improvement. And last but not least, we are driven to understand the world. That's what we want to do. We want to do what I think all of us have dreamed of, and that is understanding nature, understanding the world around us. And that's why you'll find basic and applied research, mostly in academia. So what about the government now? When you look at the government is a little different. It represents the national vision and the motivation of the country in which it finds itself to govern. And then it allocates resources. So the whole process is to decide how much for education, how much for health, how much for academia, how much for research, whatever. And the key there is to have a governance system that, in fact, represents the interests of the public. And it does it by either regulating economic activity, the law, the police, the security, and it does that by protecting public interests, whether it be defense, health, or education. And last but not least is industry. So what is industry all about? Industry is really the part of the system that assembles the human capital that was created in the university, assembles the physical capital, and also follows the needs of the market, if you will, for creating value for society. And that operationally is really the big part of transforming what you learn in the university, what you regulate in the government, into real economic value. And you need to do this, but you need to do this profitably, not at loss, because otherwise you cannot grow. And the industry is under enormous regulatory and competitive pressures. So I was head of R&D for a major company. I can tell you, regulatory is the #1 driving force for what you do, and obviously competition. So when you look at industry, industry actually does not invest much in basic research, in discovery research. That's what academia does. Industry invests primarily in what you would call late applied research and development. So that's very important to understand, because most of the industry is really torn between short-term results, profits, all of the things that you see in industry, and long-term. And you see companies that were famous that have disappeared. And that's why I always say the lifetime of a company is about 25 to 75 years. Look around you. Look at airlines. You don't remember TWA or these, they're gone. You look at many companies, Westinghouse, they're gone. Even General Electric, which was a major, major company for 125 years, is in deep trouble now. Nobody knows if it's going to survive. When you look at governments, it's a little bit longer, it's a little bit stronger than a company. It's about 50 to 200 years. If I ask you the question, how many governments since 1900 have not changed their governance? How many of them? 2. US and UK. And UK is going to change now, so with Brexit. So it is not stable as much as you would think. Every government in Europe and around the world has changed one way or another a constitution. What about universities? Well, when you look at the history of universities, you come to a 500 to 1,000 years lifetime. Actually, I just noticed from Rector Müller, 1365 means you're 654 years old. You don't look it, but you are. And you have 346 more annual days in front of you to make the 1,000 mark. But there are universities that have been around for 1,000 years. So academia has this perennial strength of being able to represent over time the values of a society, and that's why I think we need to understand the different roles and why they are important in the advancement of a society. Having said that, let me now go to give you an idea of the world as it is in science and technology. And if you look at the world today, what you will find is that the epicenter of science and technology, where about 38% of all science and technology in the world today is done in Asia. It's not done in the US or Europe. 38%, if you combine China, Japan, Korea, that's about where it is. If you look at the North American activity in science and technology, it's about 28%. And Europe is about 22%. Now, the thing that I want to tell you is that we have actually accelerated in science and technology over the past 20 years in an enormous way. If you look at the 2000 global spend in R&D, it was $720 billion. You know what it is today? $2 trillion. It's almost triple. So the whole world has really pushed for science and technology. And we're at a wave of innovation that I don't think we have seen before. And I think that's the message I want to send here, that in fact, following these trends is very important. So one of the things that we look at as policymakers, and I'm a science and technology strategist, we like to see what the research intensity of any country is. So research intensity is essentially the percent of the gross domestic product that is dedicated to R&D. And if you look at that, the highest country here at the top here is actually South Korea. And if you look at South Korea was in the middle of the pack until about 2000, and then it went and accelerated and doubled its research intensity. And the reason from my friends in South Korea, who I know, was the realization that China was rising as a manufacturing power. And it was going to do steel, and was going to do all the things that South Korea used to do. And so the South Korean government decided that they were going to invest in high-tech very advanced technology, including life sciences. And so if you look at Korea today, Korea is one of the powerhouses in bioprocessing, which it did not exist in 2000 in Korea. And then you can see China here, which was at 0.6%, between less than 1%, which is now at about the 2% margin. And Korea is about 4.5, a little bit less than 4.5. And you can see the other countries have sort of gone a little bit at a steady pace. So those are the two that have really changed. Austria, I checked from public databases, and it's about 3.16%. So it's actually high compared to the rest of the country, or the countries. And so I went to the OECD database. And in the OECD database, what you see is that Austria is actually the number 6 in the world in terms of research intensity. The highest two countries is obviously Korea, 4.2, and Israel. They are the highest in terms of research intensity. And then you can see the rest of it with Czechoslovakia, Sweden, Japan, and Austria right here. That's important because it tells you what the strategy of the government and the industry is, because they're the main funders of R&D. Academia does not fund R&D, it receives funding. But if you look at the R&D expenditures, and you say, how did we get from a world where the US and Europe was dominant, and now you have Asia that has risen to become an equal, if not greater, pole of activity that any university, including this one, need to pay attention to. And you see that very simply. If you look at the US between 1981 and today, the budget multiplied by 2. Since 2000, sorry. I took the 2000 year as a base. So you can see it's about twice. You can see the European Union also sort of went by 2. You know what the factor was China was between 2009? A factor of 20. So they obviously did not start from a big base, but they intend to really catch up with the rest of the world, and they invest in the different areas where I think we have an interest as a globe to work with, because diseases know no frontiers, and we have to work together to improve health. So if you look at that, you say, where is Austria in all of this? So let me show you where Austria is. So if you look at the totality, and this is what we call a parameter called purchasing power parity. So we're not just comparing dollars and dollars, we're comparing purchasing power. And then we're converting that into dollars. So when I give you a budget, it's really equivalent to what you can buy, the equivalent in dollars of what you can buy within that country. So obviously you can buy a lot more in a country that doesn't have high income levels, and a lot less in a country that has high income levels. But if you pay attention, you see that the US is still the #1 spender in terms of research and development at 543 billion, but China is right below 500. So we predict that within a few years, China and the US will be equal, or China will be more, because in China, the purchasing power is greater than it is in the US, simply because of cost of living differences. Germany is quite a strong country with 131 billion, and here's Austria with 15 billion. So you can see the top countries. Denmark is a little less, about a little less than Israel and Sweden, and you can see where you are in that column. And I looked at the data from the size of the country, population, and you're about 9 times smaller than Germany in terms of population, in terms of GDP. And so if you multiply 9 times 15, you come to about where Germany is. So you have about an equivalent, at equal proportion, intensity of spend as Germany does. And look at the other countries. I mean, Korea and France are less than what Germany does. So it gives you an idea of where you stand in relative terms. But that's not all you need to know. You need to know what is the direction of the country. So what I do is when I look at that, I look at the acceleration in spend, at what rate is the country investing more. And I'm not talking about just academia here. I'm talking about everything, government, industry, and academia. And so if you look at Austria, here is the top winner is China. 100%. It doubled its budget between the 2011 at purchasing power in 2017. This is where I have data. So it doubled at 100%. Austria is at 50, actually. The only countries that are higher is Israel and Turkey, for whatever reason, because of the depreciation of the money, it has a greater purchasing power now than it did before. So it's really interesting to me that, in fact, when you look at the average is about 25%. So when you look at the UK or the US, you're in the low 20s, Denmark and so on. That's where most countries are. So you're going about double the rates of the main countries and half the rate of China. All right? Gives you an idea. Then you say, okay, but that's obviously related to the human capital that works in R&D. So how much human capital are you really using to work in this R&D and science and technology environment? And so when you look at that, you find that in China, this is the number of researchers that are full-time equivalents, okay? So I'm not counting one person who works half-time. It's really a full-time equivalent. And if you look at that, you find that China has 3.7 million researchers today. You see the US at 2.3 million. Germany is at 422,000. And you are, Austria, at 50,000. About 9 times less. Then, I mean, if you multiply by 9, it's almost the German number. So that was interesting to me. But then what was more interesting is not so much the number of people who work, but the number of people relative to the number of people employed. How many researchers really, for 1,000 employees, how many top-notch, and what we mean by researchers, we mean really PhD level, doctorate level, you know, engineer level individuals. And if you look at that, you find that Austria is the number 2 country in the world after Korea. Korea has about 17 employees in R&D per 1,000. Austria is about 16. France is here, 14, 15. You can see Germany, 14. And you'll see why. But look at China. China actually has a lot to grow. Because even with 3.7 million people, it only employs about 5, 6 researchers per 1,000 employees, people who work. Interesting statistics, but then you say, all right, that's interesting, Elias, but what does that mean? How intensely these people are working? I mean, are they really doing a lot of work at these full-time equivalents? So you calculate that, and you find that in the US, you spend on average $215,000 per year per researcher. And that's all sectors, right? $215,000 is a high number, but it's driven because life sciences in the US are a big part of the spending, and life sciences are expensive. But it's still $215,000. If you look at Germany, it's a little bit below 200. Japan is about 180, 80, something like that. And then you see Korea, and then the 5th one is actually Austria with 150,000. And then when you go down, you see China, again, spends about half of what the US does per researcher. It gives you a sense of the relative position of different countries in different regions. But then the question becomes, where is this money going? How much is going into basic research, the thing that academia wants to do? How much is going into early applied research when we have a startup company that does prototyping? And how much goes to development, product development, improving cars, improving airplanes, doing research on new process in engineering and medical imaging or whatever? And what you find is very interesting. If you look at the US, here is the share for basic research, about 17%, and 19 for applied, and 63 for development research. In fact, when you look at the world, what you find is about 2/3, just remember this rule, 2/3 is development research. When you try to develop a drug or treat a disease, about 1/3 is between basic and applied. So 15, 13, 17%, whatever. Those are the general proportions. And you can see Austria, oops, sorry. You can see Austria here. So I did the calculation. I used the OECD and the National Science Foundation numbers. And what you have is a little different than the general averages, you have about 18% in basic, 35% in applied. So this tells you that you have a very vibrant early company, startups, new technologies, people who do ideas in innovation, essentially, a lot of early innovation. And then 47% is basically industrial research. If you look at Denmark, it's the same country type, 19, 37, and 44. Now, I'll tell you that one of the bigger differences between development from one country and another country is defense research, military research. So countries that have a lot of military research tend to have a higher percentage in the development area because they have companies that develop airplanes and so on and so forth. And so when you look at this, you realize that China is a little different because China only spends 5% of its research in basic research, in discovery research. And you can see here about 20% in applied research. This is why they're funding a lot of young companies, biotech companies in China. And then 84% in development, 84% in development because they have a lot of catch-up to do from all the industries that they are involved in. I hope this is helpful, but then the question is going to be, but who does this research? How much does academia do? How much does industry do? And so when you look at that, you realize that about 68% of the research is done by industry worldwide. And it varies, as I said. About 15%, Rector Miller, 15% for academia. That's what you do out of the total number that I gave. About 15% is done by academia. And then the government itself, in many countries, does research. And so government also plays a role. That's who does the research. You have national laboratories that are owned by the government and so on. But then you say, who funds it? Who gives the money? And you realize that industry actually funds itself a little less than what it does. So they do receive subsidies from government, usually, to do R&D. And then you can see government provides about 30%, anywhere between 25 and 30% of the funding. So I hope I'm not boring you with these details, but they are very important, in my view, to understand where is the money, and then where's the output. So one way we measure outputs is patents, international patents, patents that are filed with the PTC office, which is a worldwide office for patents, and you see that the highest producer of patents is actually Japan, with about 60,000 patents a year. And in Japan, it's part of your promotion. If you are an employee in Japan, you file a patent, you get a bonus. So there is a big push to create patents in Japan. And then you see the EU is second with about 56,000. Then you see the US, about a little bit less than 50. Germany, about 23. And Austria is about 2,000. So if you multiply by 9, as I said, you're a little bit less than Germany in terms of producing patents in Austria, but that may be because you really did not accelerate until the 2010, 2011 in terms of investing in R&D. So I just want to finish this part of my talk by drawing some conclusions. So the conclusions are, number one, R&D activities have tripled in the world since 2000. There is a real investment and real knowledge that is generated. The regional balance has changed. Asia was not as strong, and now it's Asia, 38%, America, 28%, and Europe, 22%. Austria, clearly, from my analysis, has accelerated its R&D intensity since 2011. And when I look at all the indicators, Austria is in the top 10. So you have a good base for this university to be here 1,000 years from now, I hope. So the key thing here is we're talking about life sciences, so how much of this is life sciences? It's about 25%. Everything I told you, about 25% is spent in life sciences. So then the question is, where is life sciences? Where is biomedicine today? And so I'd like to say to you that we've seen enormous progress in knowledge, in technologies, in advances, in every part of life sciences. But then we're very unable yet to control diseases. We're not very good at that. And so the question is why? So that's what I'd like to address in my view of why we have a problem and therefore where we need to go. First and foremost, as we discovered more and more about the structure of biological systems, we realized the complexity is much greater than what we thought. In the 70s, people thought that there would be one treatment for cancer, magic bullet. Today we know that cancer is 1,000 diseases. Then precision medicine is needed because we can no longer afford to do a one-size-fits-all treatment for everybody, regardless of the efficacy of the treatment. That's what P4 medicine is all about. And then the scale of the problems as we see them today, when I was at the NIH, the number one thing I tried to do was to break barriers between disciplines, because you realize that you cannot solve the problems we have today with a very reductionist approach. You really need an integration of the whole team working together. And that means interdisciplinary culture, interdisciplinary structures. And I've created, when I was in the US, I've pushed the NIH to create a lot of interdisciplinary institutes. It's called Institutes Without Walls. So you see multiple people, from physicists, from engineers, working together at the bench, and this is why we created these grant programs that are not just for doctors or for engineers or for physicists, but they're for all three together, working for one project. And the last thing that we noticed is the convergence of sciences is enormous. When you see structural biology, which comes from real physics, and you see the structures that come out of that for biological molecules, you realize that the convergence of science between the biological system and the physical system is happening at a very high speed. And last but not least, we have to realize that we're not good. 90% failure rate in industrial R&D for therapies. We fail a lot. And so I also conclude that we need much more basic research than we have until today. So let me just go a little bit faster now by talking a little bit about the complexity of biomedicine. When the university was created in 1365, doctors at the time looked at the organism. They wondered if you had fever, if you had a bad air in your house, and if you're red, and then if you were, they bleed you. That was it. They looked at the organism, right? Today, we don't have that view of the world. We have a very molecular view of the world, starting by the cell. So as you look at it and you try to represent that in a mental way, my representation is like this. The world is made by a cell, which starts, from one cell and then divides and divides through DNA, RNA, proteins, all that. And then they create tissues. And these tissues create organs. And these organs eventually create an organism, which you are, through billions and trillions of divisions and mechanical interactions that lead to an organism, which then expresses behaviors. So your behaviors are emergent properties of your organism. And that's affected by the environment in which you live in, which means that every single one of us fundamentally cannot be like the next person. I mean, your fingerprints are different, your eyes are different. I can also tell you that your brain is very different from each other. Even twins, when you look at the genome of neurons in twins, you find that there's a 40% difference in genomic comparisons between neurons of homozygote twins, which means that this complexity on one side leads to an individuality, that each one of us is a unique individual. So how do we really solve this conundrum, and how do we define a strategy for science was the question I was really fighting with and trying to figure out. And I've come to the conclusion that if you look at the systems that we're dealing with, they're multi-layer systems. And if you really want to represent that in your mind, I made a cartoon for you. And if you're on the left side here, you look at DNA, and you say, okay, how does this all work? You realize that all layers are interconnected and interrelated. So we know that DNA does not do the whole work. There's epigenetics. And then the code within the DNA defines genetics and genomics. And then you know that you have to translate that into proteins by transcribing it into RNA. So you have another layer of complexity, transcription and translation. And then all of these things are interacting with each other and are regulated by each other. in ways that we do not quite understand. And so when you have, for example, a mutation in a particular protein, that protein is part of a pathway. And that mutation can either enhance the activity of the pathway or reduce the activity of the pathway, and will make changes in the observable metabolics and proteomics. So you might have a high cholesterol level or another marker. That essentially is what I call the Russian doll concept, where you see the doll inside the other, except that the layers are talking to each other. And so when you look at the complexity of that, you realize that even our best computer systems today cannot capture the complexity of the interactions, and that's where the challenge is. Add to it the fact that the environment also affects these multi-layered interactions. And so that, to me, is the fundamental aspect, because the environment and the nature of biology are really where I think we face a great challenge. Why do people become obese? Because the environment changed. And if you don't believe me, I have an experiment here I'm going to show you that proves that the genome does not drive diseases. It doesn't by itself. Environment does. So we had the statue of David, you know, from Florence, and that has no DNA. I can tell you, we looked, there was no DNA. And we took it to the US for 1 year. And that's what happened. So you can tell that the environment truly has an impact on our biology. So what is the time? So I can summarize this for you in saying that we're going from a period where all research was reductionist. We try to understand the hardware of life, proteins, DNA, and all. And now we're going to the software of life by understanding in a quantitative way the network biology that I just described. And so that understanding will lead to a reclassification of diseases. You know, when you celebrate 50 years from now your university day, I guarantee you the departments you have today will not be the same, because the diseases will have changed, because their definition will have changed. This morning we heard in the lecture that diabetes may actually not be 2 diseases but 15 with different patterns. That knowledge is going to accelerate as we understand that, but it will also allow us to define biomarkers which are predictive, personalized, hopefully preventative, but will need the participation of the patients. We cannot have patients who say, oh, I want my privacy, I don't want to participate, but then they want to benefit from this new medicine. So socially, the social sciences are going to have to help us make sure that this new medicine is participated in by the patients, because hopefully in a few dozen years, patients will not be sick when they see the patient, when they see the doctor. They won't just wait until they get sick. They will be treated before that. So that really leads to a better understanding of the environment of the patient, and that requires large data integration and analysis of populations through these novel technologies that are coming up, which are very important. So to me, from my point of view, when I was head of R&D, I said, you know, if indeed we are reclassifying diseases according to their molecular networks, what we realize is that no single disease is simple. No single disease is due to a single target, except for monogenic diseases. So you always have interacting networks that play in the disease. So for example, you can develop a drug for atopic dermatitis, which I did, and lo and behold, it works for 65% of the people, and it doesn't work for 35%. And you say, why? Well, why is because you probably have an alternative pathway to the disease that you have not addressed. And that's why I say the future of medicine and the future of pharmacology is going to be multi-targeted drugs, multi-specific antibodies or others. Once you have really mapped out the molecular pathways, then the tools are there. We have developed the tools. I mean, you have RNA, you have small molecules, you have multi-specific antibodies, which is what I worked on, multifunctional peptides, and something that, you know, has been empowered by something that happened here, CRISPR-Cas9, with gene editing as cell therapies. So that's the future in my view, and the emerging translational paradigm, something that universities like yours are trying to do, is to, on the one hand, what I call on the left side, is what I call multi-layered biotyping. It's not genomic, it's biotyping, because people always talk genome, phenotype correlations. I don't think that's enough. You really need to look at that multi-layered process from epigenome to genome to transcriptomics, even immunomics, and build that at the atomic level in models that even today can be done at a single cell. And this multiplication of interrelated data is absolutely necessary. No university that I know of has really reached that stage today in the world. Many are fighting, many are trying. I mean, I know in Boston there's a large project with partners. Geisinger is sequencing all their patients. But when you look at it, you find that no one has been really comprehensive on this. But you need to have a population around you to be able to do that. On the right-hand side is what I call comprehensive phenotyping. In other words, with the new methodologies, you can truly study what we call extreme phenotypes. People who have very extreme form of a disease, because then the genetics can tell you the target that you want to address, or longitudinal population studies, which have always been very rich, like the Framingham study for cardiac disease. Biomarkers are going to be essential. If you don't have a very strong pathology department, chemistry department, microfluidics, all of those capabilities of what I call biological analytical skills, you cannot really correlate with the biotyping and with the patients. Solid and liquid biopsies, they've already revolutionized the world of pregnancy, for example, or detection of trisomy 21. Human genetics, real-time biosensing. We can no longer wait for a patient to come every 3 months and tell us what happened. They can't remember. So you have to sense the patient real time. You have to have health records that mean something, not just a hodgepodge of data. And last but not least, imaging, which is my love and what I've done. So I'll talk to you about the role of imaging in this context, in my view. So none of this can happen, however, without a very strong data sciences capability. And the thing that I want to share with you is that I feel that medical schools and medical universities are very shy about data sciences. They think it's not theirs. They think it's a computer department. It's the IT department. No, no. A data science skill is going to be as important as a stethoscope. You need to be first class in data sciences. And don't think that others are going to do it for you. So get your best, smartest people, recruit the best doctors who also have data science skills, and get them into the medical school. That's my message. So, imaging, I will tell you, imaging really plays a big role in this phenotyping of the population and understanding these particular networks at both ends, at the molecular level and also at the full population level. So at the molecular level, I think there's a revolution because we need to assess functionality of the therapeutic molecules in vivo. We need to obviously characterize the response of a disease. Is the treatment working, not working? Why? And then we need to have multimodality imaging because there's no one source of imaging that can really give you all the information you need. So I will show you here work that I did when I was at Sanofi when we were looking at an antibody called against TGF-beta. And we had good evidence that in patients who did not respond to immuno-oncology drugs, they had a high TGF-beta, which is basically an immune suppressor. And we found that, in fact, the penetration of T cells in the tumor was dependent on TGF-beta. But we needed to prove that. And Dr. De Vries in the Netherlands, he's one of the leading doctors where she can take an antibody and she can tack to the antibody a radioisotope, which has a long half-life, zirconium 89. And so in this particular case, we had the ability to show whether or not TGF-beta was in the target tumor or not. And we thought that that was important because if it was, then our therapy could work because the target was there. The second example I would show you came from Johns Hopkins, from Marty Pomper. You know, the prostate-specific membrane antigen is very specific to prostate and prostate cancer. So he had the idea of finding a molecule that is very specific to that PSMA. So he created the second-generation molecule, which allows you to create a radioisotope with fluorine-18. And by doing that, he was able to show, in animals, but also in humans. So on the left-hand side is a prostate cancer patient with a non-specific bone scan. On the right-hand side is with the specific covalent binding to the PSMA, to the prostate-specific antigen. And you see the difference between the two. Much more metastatic disease on the right. But then he had another idea. You know, what is an issue in modern medicine is you act on the body, but you don't know really what's happening. And this is really important in cell therapy. So when you use cell therapy like CAR T cells, you really don't know what happens inside the body. Does it go to the tumor? Does it not go to the tumor? So the challenge was to image in the body these cells. So what did he do? He had the idea, because for those of you who may not understand what CAR T cell, CAR stands for chimeric antigenic receptor. So the gene engineers with gene editing, including CRISPR-Cas9, will put an antibody on the surface of a T cell, which is specific to the tumor. So that antibody will bring the T cell to the tumor, and the T cell will kill the tumor. That's the idea. So what he had the idea, he said, well, for each T cell that is therapeutic, he's going to put a PCMA gene inside of the T cell. So instead of one gene, he puts two genes. And the PCMA gene, the PSMA gene, will be for imaging, and the CD19, which was his experiment, CD19 was for killing the tumor. But that killing cannot happen unless the cell goes to the tumor, right? And so he did that, and look what he showed. He showed on the T cells, you cannot image the T cells per se. Then you have the tumor, and this is the T cell imaging at day 12, and he can show that the tumor has been infiltrated by the T cells. So that's very important, and I think it's going to be the future. The other part of imaging that is going to revolutionize medicine is what we call radiomics, where you're going to phenotype not just a patient at a time, but an entire population by making correlations. And I think it's probably the field that is the most advanced, because images can be analyzed in a very formal way, just like face recognition or iris recognition. The same thing is happening in Chris Harrell's department. I was visiting today. They were doing this for the liver. And I think it's happening around the world. And you're going to see that this means integration of healthcare systems. In other words, you cannot do it with just the University of Vienna. You don't have enough patients to do that. But you will have to collaborate, maybe at the European level, to create reference databases, where in the future, the radiologist is not going to just read an X-ray and tell you, well, I see this or that. No, that X-ray is going to be, or CAT scan or PET scan, is going to be compared to maybe 5 million MRIs or CAT scans. And then the thing will come back and say, this particular patient looks very, very, very much like this particular patient or this group of patients. So it's like face recognition on a scale we've never seen before. And so if you look at this, you can track the evolution of diseases, because you can have the same patient come back and be compared against its own self and against another population. And so that's novel information we've never had before. We always dreamed of this, but it's happening. And when I was at the NIH, I realized that this was going to be something important, even though we didn't have all the technologies. We started a study in 2003 with industry and with NIH, called the Alzheimer's Disease Neuroimaging Initiative. What we decided to do was to take patients with family history, potential for Alzheimer or early Alzheimer, and we followed them over time. We did imaging, and we did also a sampling of biomarkers. It's probably the most successful biomarker study, simply because we're now able to analyze the images, and I can tell you the changes that occur in very detailed brain surfaces and structures of the brain, I would think are going to be very predictive of which group of patients is most likely to develop dementia. So that is a revolution. That's something we've never been able to do, because these changes are very subtle, but what's important is the change from year to year to year. So these patients are imaged every 2-3 years, and we see the changes in the brain. So that on the right is actually the mathematical representation. Each dot represents the change over time. Then we created also a database of young children with MRI through life. So this is a cohort of patients started again in 2003, 2004, which is continuing today, which tells you exactly how the child's brain develops and what happens when the disease comes up, which will be a reference database. So imagine that you can scale this, and you could do this for every patient. You could store the image, large database, use machine learning to predict and identify new diseases. So, in conclusion, I hope I've been able to tell you that, number one, the world is accelerating its commitment to research. Number two, the tension between complexity and precision will force these two fields to come together, which will create a convergence, in my view, between diagnostics and therapeutics. Why? Because if you are able to characterize a target diagnostically, you can treat it. We have the tools to do that. But we don't understand enough what exactly happens in diseases today. So that's the next century of effort that we have to have, and maybe the next second century for your annual day. And because, you know, in science, to me, what I always tell everyone, what is the greatest risk in science is to not take a risk. That's the greatest risk. And the second thing I tell my students, I said, you know, what makes the difference between a great surfer, you know, the surfing on the wave? What's the difference between a great surfer and not so great surfer? You know what the difference is? It's picking the right wave. If you have a great wave of science and you ride that wave of science, you'll be successful. So I wish you, Mr. Rector Miller, and all of the faculty here, great success. I think you're clearly accelerating as a country and as an institution. And so I offer you my congratulations for your first annual day. Thank you very much.